Hi everyone, it's Dr. LeBlanc with Unit 6, the big picture of regulating eukaryotic gene expression. What's depicted below is the life cycle of a frog. A frog starts out as a zygote and undergoes many different morphological changes until you become an adult frog. At each stage and all along this process of development, genes are expressed differentially. They're turned on at the right time, in the right tissue, and at the correct amounts in order to end up with an adult frog from just a fertilized egg. Ever since scientists discovered that every single cell in an adult organism contains a copy of the entire genome, how genes are regulated has become the holy grail of molecular genetics. Differential gene expression is one way to express the processes that occur in embryonic development. This is a very interesting link about human development. It's about 8 minutes, but I highly encourage you to look at it if you're at all interested in developmental biology or if you're interested in how you became a human. Frog development is a model system that many developmental biologists and geneticists study. Let's take a look at this remarkable process. We start out as a fertilized egg that divides. During the blastula stage, every single cell starts to divide and divide and divide and becomes smaller at each division. This is the blastula stage. At the end of blastula stage, sheets of cells start to move around to form what's called a gastrula. During neurulation, neural folds form on the surface of the embryo, move toward each other and fuse to become the spinal cord and brain of the tadpole or eventual adults. From this point on, the embryo undergoes morphological changes, changes in shape, as well as changes in gene expression. You develop eyes and a gill and a tail. And eventually they hatch and become a tadpole. During the tadpole stage, they grow legs and eventually turn into adult frogs. Drosophila melanogaster, or the fruit fly, is also a very well-known model system for embryonic development. During embryonic development in Drosophila, a series of cascading events occurs where maternal effect genes, which are transcription factors, activate gap genes, which are also transcription factors, that activate pair rule genes, that are also transcription factors, and segment polarity genes, which are also transcription factors. So it's one big, long cascade of turning genes on that are transcription factors. Eventually, you turn on the Hox genes. Hox genes determine the identity of segments. In other words, that these blue segments are going to produce wings and legs, and these segments are going to become part of the abdomen. The order of gene expression from anterior to posterior parallels the order of genes on the chromosome. So in the Drosophila genome, all of these Hox genes are arranged in the same order in which they are produced in an anterior-posterior pattern in the embryo. Let's look at one particular gene for a transcription factor called even-skipped. Here is the even-skipped gene. Here's the start site of transcription. Here's the structural region of the gene. Surrounding the gene are several enhancers that dictate its expression in a particular region of the embryo. This is an expanding view of an enhancer that turns the even-skip gene on in stripe number 2. Notice that there are binding sites, many and overlapping, for activators and for repressors. Combinatorial control determines which and how many transcriptions are bound, determines the fate of transcription. One gene can have many sites for both activators and repressors. So let's take another look at even skipped expression in the embryo. This is how even skipped is expressed. It's expressed in 7 stripes throughout the embryo. So how do you get expression of a transcription factor in just 1 stripe of the embryo? It's by combinatorial control. So if you notice, in this region of the embryo where even skipped is expressed, right down here, activators that can bind to these positions on the enhancer are present. Bicoid is present in this region, and so is hunchback. But what's missing from this region are the repressors, cruple and giant, not expressed here. Okay, and notice that at the borders of the stripe, where even skipped is not produced, you see the presence of the repressors. So differential gene expression in embryo can explain what goes on during embryonic development. We can also have differential gene expression in response to environmental stimuli, such as the immune system. Your immune system mounts a response to a foreign invader, and the expression of genes in those cells changes. Liver toxins, if you eat Fruit Loops for breakfast, all of the bad food coloring that's in the Fruit Loops has to be dealt with, and your liver will do that and change the expression of proteins that are going to clear the bloodstream of those foreign substances. Hormonal cycles obviously change gene expression, as does diet and physical activity. So the overview of the regulation of gene expression: we have transcriptional level control, which we just saw. We have control at the level of RNA processing, alternative splicing. We have regulation at the level of translation via small RNAs, microRNAs, and siRNAs. And we also have regulation at the level of post-translational modification, including feedback inhibition and covalent modifications such as phosphorylation that regulate protein function.